Chapter 4, it's about genetics and cellular function. Towards the end of this video, we will go over the key points for Chapter 3 in Chapter 4. So let's start with what is DNA. So DNA is deoxyribonucleic acid, and this is a macromolecule that it is located in the nucleus of the cells. DNA has the structure of a long thread that has a uniform diameter of 2 nanometers, and the length of the DNA, it varies. Our DNA, it is distributed in 46 chromosomes, and most of our cells, with the exception of the red blood cells, have a nucleus. So that what it means is that they have DNA, and within this DNA we have, as I said, this distribution of the DNA molecules into 46 structures known as chromosomes. The average DNA molecule has about 2 inches in length. Now the DNA is made out of several monomers known as nucleotides. And when these nucleotides bind to each other, they form a long polymer that we know as DNA. Now the nucleotides have 3 units. They have a sugar, that it is a ribose sugar. They have a phosphate group, and they have a nitrogenous base that, depending on what type of nucleotide you are talking about, it will be a single ring nitrogenous base or a double ring nitrogenous base. So the name of the sugar actually for DNA is deoxyribose. So it's a ribose sugar, but it lacks an oxygen. That's why it's called deoxyribose. And then into one of these ends of the deoxyribose sugar, we have a phosphate group attached to them, and then we have this nitrogenous base that it can be a purine if it has a double ring, or it can be a pyrimidine if it has a single ring. Now the 2 purines that we have in DNA are adenine, which is abbreviated A, or guanine, which is abbreviated G. So these 2 will be purines or double ring nitrogenous bases. Now the pyrimidines, they have a single ring, and they are named cytosine, which is abbreviated C, and thymine, which is abbreviated as T. Now the cytosine and thymine are found in DNA, while uracil, which is another pyrimidine, it is found only in RNA, and the DNA bases then are A, T, G, C, and these pair in a specific manner. So adenine always pairs up with thymine, and cytosine always pairs up with guanine. And because they have this double ring or single ring, then depending on what base you are talking about is how would you classify it. Now specifically uracil, which is found in RNA, is substituting thymine in the DNA of in the structure of the DNA. So DNA doesn't have uracil, only RNA, and RNA has only adenine, uracil, cytosine, and guanine, so it doesn't have thymine. So this is the structure, the figure showing you the nucleotide. So this is adenine, it's a purine, so it has a double ring nitrogenous base, which is this, and then this base, it is attached to one end of this deoxyribose sugar. So in here, this ribose sugar doesn't have an oxygen in this part, so that's why it's called deoxyribose. And then in here, this ribose sugar or deoxyribose sugar has attached this phosphate molecule. So this is how the nitrogenous bases then will look like. So these are the purines, adenine and guanine, and then this will be the pyrimidines, cytosine and thymine, and then uracil for RNA. This thread-like structure of DNA, it forms a double helix, and it looks like a spiral staircase. And on each side of this double helix, you have phosphate groups alternating with the deoxyribose sugar. And then in between the two backbones are the pairs of the nitrogenous bases, and these nitrogenous bases will be on each strand of DNA, because it's a double-stranded structure. You will have base pairing of the nitrogenous bases by having hydrogen bonds. So a purine on one strand always will bind to thymine on the other strand. And adenine and thymine will have 2 hydrogen bonds, and cytosine and guanine will have 3 hydrogen bonds. So if you want to break up the bonds between adenine and thymine, or between cytosine and guanine, the one where you need to provide more energy to break the bond will be between cytosine and guanine, because they have a triple hydrogen bond. So this is a much stronger than a double hydrogen bond. Now then, this is the law of complementarity of base pairing. So if you have an A on one strand, you always pair it with a thymine on the other strand. And if you have cytosine in one strand, you will base pair it with a guanine on the other side, or in the other strand. So one strand determines the base sequence of the other. At the atomic level, that's how these molecules of DNA will look like. So it looks like a spiral staircase in which you have 2 strands. And then if you see a representation of the hydrogen bond between the 2 strands, you will see that between adenine and thymine, you have 2 hydrogen bonds, and between guanine and cytosine, you have 3 hydrogen bonds. And that's always the same. So then in between the strands, you have the nitrogenous bases, and then in the backbone, you have the deoxyribose sugar and the phosphate group. Now, those sequences of nucleotides that are located within the DNA form segments, which we know as genes. And these genes, they code in many cases for synthesis of a specific protein or for other instructions for the cells. So if you count all the genes that exist in one person, that will be the person's genome. In general, humans, we have about 20,000 genes, and only about 2% of they make up only 2% of the total DNA, and the other 98% is what we call the non-coding DNA. In the non-coding DNA, we have structures that are important for regulate the gene activity and to actually produce many varieties of proteins. Now, the DNA, it is also known as chromatin, and chromatin is a fine filamentous DNA material that it is packaged with proteins. We have then this DNA found in 46 chromosomes in most of the cells, with the exception of sex cells in which we only have 23 chromosomes. And if you elongate the DNA material from a single chromosome, you will have 6 feet of DNA that it is packed in the cell nucleus of about 5 micrometers in diameter. Now, there is these cells that sometimes are non-dividing, in which the chromatin is very slender that you cannot see it with the aid of a microscope. And then if you use, with the aid of a light microscope, if you use an electron microscope, this chromatin will have a granular appearance. So this is how the chromatin looks like. Chromatin, it's another way of saying DNA at some point. And this is the actual most deep part of the DNA. So if you elongate one of these, and if you extend one of these chromosomes, which is this one in the bottom, you end up looking at something like this. So this is the DNA double helix, so it has one strand and another strand, and then you have the bases in between, which makes like the rungs of a ladder. And if you see here, from edge to edge of the double strand, you have 2 nanometers in diameter. Now, if you go to the next step of the formation of chromatin within the chromosomes, you find out that this double-stranded DNA will wrap around these proteins that we know as histone. And there is this wrapping around the DNA that when you just do like 2.5 turns around one of these histones, you will extend one part of DNA between 2 different histones, and this will be called linker DNA. And this structure is going to be 11 nanometers in diameter. So basically it's a little bit over 5 times of your diameter of your double-stranded DNA. Now this structure will wind up a little bit more and form a 30 nanometer structure, in which these nucleosomes, which is basically part of the linker DNA and the core particle that wraps around the histone, and then you wrap it more, you'll form a 30 nanometer fiber, and it looks like a zigzag. And if you keep wrapping it around, you will form a 300 nanometer fiber, and this one will form like irregular loops, and then you keep wrapping it more, and then you form a 700 nanometers fibers that it is going to be further coiling until you form these actual complete chromosomes. Now the chromosomes have these 2 structures that are called sister chromatids, and they join at the center of the chromosome, which is the centromere. So what are histones? These are proteins that are crucial for the packing of the DNA. Histones are made of several amino acids that have a positive charge, and because the DNA is negatively charged, it tends to wrap around these histones. Now the histones are made out of specifically 8 molecules, and then the DNA, when it's wrapping around, it forms like a spool, and then the chromatin will be consisting of thousands of repeating nucleosomes, which will be the DNA wrapping around these histones. So what is the nucleosome consisting of? A core particle, the histone cluster with DNA 1.5 turns, it's actually like 2.5 turns, and then the linker DNA, it's a short segment of DNA that connects one core particle with the other one, and then you form this zigzag-like structure, and the chromatin will be thrown into a complex of irregular loops and coils, and it will form 1,000 times shorter than the original molecule. So when you have these 46 chromosomes then within the nucleus of the majority of the cells, you distribute these chromosomes into a particular area or chromosome territory within the nucleus. And these chromosomes will be close to the channels of the nuclear membrane that allow it to have chemicals coming in and out of the nucleus, so that you can have easy access to these chromosomes, to these genes within the chromosomes, and then you can regulate the activity of the cell. Now, you can think about if the cell is not dividing, because cells tend to divide to either repair damaged tissue or to grow. The cells tend to divide. So you can think of that only the DNA is very active when the cell is dividing, but actually, even in a non-dividing cell, the chromatin is not static. It will have changes, periodical changes from time to time, according to what is the genetic activity of the cell. And during this genetic activity of the cell, some of the genes are turned on and off. And we have some examples. For instance, when we are developing, some chromosomes or some genes within the chromosomes are more active in some cells than others. For instance, if you are going to make liver cells, all our cells have actually the same DNA, and they have the same genes, but in liver cells or the future liver cells, you activate liver cell genes. So when they are active, all these cells during development become liver cells. And then you turn off from these cells the genes for hair, skin, the genes for heart muscle, etc. So you only make a liver cell. So this is how you can turn on and off these genes. Now, when the cells are preparing to divide, then the cell has to make a copy of all the nuclear DNA. And the purpose of this is for having 2 copies of DNA, and you will give a copy of DNA to each of the daughter cells. And as you're doing this, you are separating the duplicated DNA into their corresponding chromosomes. And then as the cells are preparing to divide, the DNA condenses and forms visible chromosomes that can be seen under a light microscope. And then the chromatids, the sister chromatids, will join together at the middle of the chromosome, which is called the centromere. And on the outside of this centromere, you have a specific plaque of proteins that is called the kinetochores, which is going to be an important structure for the microtubules of the mitotic spindle to connect so they can separate the chromosomes. So this is how the chromosome looks like at a stage of nuclear division, which is called the metaphase. So here you have condensed the DNA, and then you have this portion of the chromosome that is called the centromere, and on the edges of the centromere you have this kinetochore. And each of these parts of the chromosomes are known as sister chromatids. So you have one chromatid and a second chromatid. So what you must do in order to have cell division? Well, you have to first make a copy of DNA. And when you make a copy of DNA, this is known as DNA replication. And this DNA replication happens during the cell cycle. And the purpose of this DNA replication is, as I mentioned before, to give a complete set of genes or DNA to each daughter cell. So usually our cells, when they're going to repair or when they are going to grow, they undergo this DNA replication and they form two daughter cells. And the actual two daughter cells will have to receive each of these a complete set of genes. So one of the purposes of replicating DNA is also to give not only the same number of chromosomes to each daughter cell, but also give the same copy of DNA. So the same copy of DNA with the same genes. So you don't get one cell with different set of genes than another. And this is given by actually replicating the DNA, hopefully without any changes and mutations. So following the law of complementarity. And then we have these 4 steps for DNA replication. So in a already condensed chromosome, what you will do is you have to unwind the chromosome from histones. So you can access the nucleotides, so you can start copying these nucleotides. Now we have this enzyme that is called DNA helicase that helps to unwrap this double helix and exposes these nucleotides. And when it's doing that, it will form something that is called a replication fork, in which you have one strand going into one direction and the other strand going into another direction. The next step will be to actually make the copies of the nucleotides by the use of one enzyme that is called the DNA polymerase. So the DNA polymerase will bind into the nucleotides and start reading what is the actual sequence of nucleotides, and it starts making this new copy of nucleotides out of free nucleotides that might be found within the surrounding areas. And then as the polymerase is extending these new DNA strands, the polymerase will separate the polymerase molecules to work on each strand, proceeding in opposite directions. And then the polymerase will be moving towards the replication forks, making a long continuous strand of DNA. And the other polymerase that is moving away from the replication fork will make short segments of DNA. And these short segments are known as Okazaki fragments. And there is this enzyme that is called DNA ligase that is going to make like a little bridge, adding more nucleotides into where you have a gap between two adjacent short segments, basically ligating these short segments, put them in, together. And then ultimately you make two daughter DNA molecules from the original template or parental DNA. And you have something that is called semi-conservative replication, in which each daughter DNA will consist of one old strand and one new strand of DNA. And then once you make the DNA, the new copy, you have to repackage this copy. And then you will have thousands of polymerase molecules working to make these copies of DNA, so you can complete your 46 set of chromosomes into 6 to 8 hours. So this replication has to happen really rapidly. And at this time also, what you need to do is to produce more histones, so you can have these histones available, so you can wrap around the newly formed DNA into these histones, and then you can have these new nucleosomes distributed in the new DNA. So this is how then DNA replication happens. So first, with this enzyme, which is called DNA helicase, you unwind this double-stranded DNA. So you separate it. And then, as I told you, one of the strands of DNA is going to go in one direction, and the other one is going to go into another direction, into what we call the replication fork. And one of the polymerase, the one that goes towards the direction of the replication fork, will form this long, linear, continuous new strand attaching to the old strand, which is this. So the one in blue is the old strand, the one in orange is the new strand. And then where does the polymerase makes this new strand of DNA? Well, using incoming nucleotides. So this will be the monomers that makes this polymer. And here within this other strand, where the polymerase goes in opposite direction to the replication fork, you have this continuous making of the new strand, and there will be a gap in replication that this enzyme, DNA ligase, is going to fill in. And then you can form a continuous strand of DNA. Since the DNA molecules are made out of many nucleotides, millions of them, there is always the chance that as the DNA polymerase is replicating these new nucleotides, it can make a mistake. So for instance, if you have in the original DNA sequence, you have A or adenine, and then you have thymine, thymine, and adenine, that little sequence in that order, A, T, T, A. What you expect that the polymerase do is to form in the new strand the complementary sequence, which will be T, A, A, T, because adenine is complementary to thymine. But what if the DNA polymerase, instead of doing this ATTA, it does one sequence in the chain of DNA, the new strand of DNA, that it is AGGC. So that's a mutation. So one way to prevent this from happening is that as the DNA polymerase is doing these mistakes, you can stop the process and try to check for these mutations. So you have these checkpoints within the cell cycle that helps to bring enzymes, repair enzymes that can correct these mistakes. And hopefully, once this mistake is corrected, the cell can continue with the actual process of cell division. Now, there are some mutations that definitely the cells cannot tolerate. And when that happens, the cell definitely stops the cell division, and then it halts the process of replication of DNA. And the cell does not continue the cell cycle. Now, what are then mutations? They are changes in the DNA structure, and many of these changes can be due to environmental factors like exposure to radiation, to viruses, and to chemicals. Sometimes we have genetic mutations as well, and depending on what area of the DNA or in what gene those mutations are located is either if you don't have an effect on the function of the tissue or the cell, or if you have a problem like the development of cancer or even death. So if the mutation happens, for instance, in an area of the DNA or in a gene that is very critical, more than likely the organism will die. So what is the cell cycle? The cell cycle is the cell's life from one division to the next. And cell cycle is subdivided into two major phases, interphase and mitosis. And interphase has three subphases, one that is called G1, another one that is called S, and another one that is called G2. Now G, it comes from the naming of these phases as they were studying mitosis early in the study of the cellular division, and they call this phase gap because apparently the cell wasn't doing anything, but actually the cell is always doing something in any phase of the cell cycle. So G stands for gap, and then S stands for synthesis. Specifically, DNA replication happens during synthesis. Now, most of the cells have G1, S, and G2, but there is a fourth phase in specific cells that is called G0, in which the cells, they just form the original copy and they enter into the G0 phase and they don't go back into replicate. The cells that do this are cells that are terminal cells, like the neurons. So the neurons, once they form the mature, the neurons, once they form their mature structure, they enter into G0 and they don't replicate. If you damage these cells, the neurons, they don't replicate again. So they don't go back into mitosis. Now, mitosis have several subphases as well. So it has prophase, metaphase, anaphase, and telophase. So G1, what happens during G1, the gap 1 phase? So is the interval between the cell birth from division and the DNA replication. In this case, the cells will carry out normal tasks and starts accumulating materials for synthesis, like it starts accumulating nucleotides, histones, and also organelles. Now, when the cell has completed the accumulation of all these materials, it will enter into the synthesis phase. It's like if you are going to build anything. What will you do in gap 1? You have to go and purchase all these materials, so you will be able to make synthesis of something, okay, or build something. Now, during S phase, then, we will have the replication of the DNA and also the duplication of the centrioles. And then after you do the replication and you check that the DNA, it is correctly replicated or duplicated, you enter into G2 phase or second gap phase. And during the G2 phase, what happens is that the cell checks again for replication errors or mutations in DNA, and the cells starts growing and synthesizing enzymes that will control the division of the cells. And then the next phase is going to be called mitosis, in which the cell will actually divide the nucleus. It's wrong in there where it says replicates nucleus. So basically, at some point out of the 40, in synthesis phase, when you have made your 46, out of your 46 chromosomes, 92, because if you multiply 46 times 2, that's give you 92 chromosomes. During M phase or mitosis, what you will do is that you will divide these 96 chromosomes that you made during synthesis into two nucleus, each nucleus with 46 chromosomes. So you, at some point, divide the nucleus and form two new nucleus. So, and then as well, what you will be doing is as distributing to the cells equal number of organelles. So it's not only dividing the 96 chromosomes into two daughter cells, each with 46 chromosomes, but also you have to give equal number of organelles. Like if you have made 20 mitochondria, so you will give 10 mitochondria to one daughter cell, 10 to the other one. And then again, G0 is the phase that describes cells that left the cell cycle. So once the cell makes its own copy, it will enter into G0, and then it won't go back to replicate. And then the cell cycle is variable in terms of time for the different cell types. Some cells in our body replicates very rapidly, like our hair cells, like the cells in our intestines. Other ones take longer for replicating. And in this figure, we can see the phases of cell division. So if you see here in green color, you have interphase. And if you see here kind of in a brown, light brown color, you have mitosis. And the cells during their cell cycle, they spend more time in interphase than in mitosis. So it takes more time to prepare the cell to duplicate than the actual cell division or mitosis. And then again, cell cycle is subdivided into G1, S phase, and G2. And there will be another one in terminal cells like neurons that goes like here into this part that is called G0. And then mitosis will be subdivided into prophase, metaphase, anaphase, and telophase. That's the 4 stages of mitosis. Now, what is cytokinesis? Cytokinesis is actually the cell division or the division of the cytoplasm into two daughter cells. So mitosis can be defined as a cell division that results in two genetically identical daughter cells. Functions of mitosis is to develop an organism out of one fertilized egg to roughly 50 trillion cells. Also help us to grow of all the tissues and organs after we're born and even before we're born, and helps to replace cells that are damaged or that they have died. So now let's talk about what happens in each of these phases of mitosis. So during prophase, in which you have the start of mitosis, you start condensing the chromatin into compact chromosomes. So you can see under the light microscope now the newly compact chromosomes, and you will have 46 chromosomes, each with 2 chromatids per chromosome. And then these chromosomes will be subdivided into their individual chromatids for each of the daughter cells. So you start with something, 2 sets of chromatids, and then you have to dissolve the nuclear envelope so that the microtubules that are being formed within the centrioles that are located within the cytoplasm can penetrate this almost disintegrated nuclear envelope, and then you can attach these centrioles into the mitotic, so you can connect these microtubules, into the kinetochore in the center of the chromosomes. Now, in metaphase, what happens is that the cells will have a center that is called the equator, and the chromosomes will align in a horizontal fashion in the cell equator into what we call the metaphase plate. And once they are aligned, these chromosomes, you will have the completion of the mitotic spindle, in which the microtubules that are connected into the kinetochores of the chromosomes will start pulling these sister chromatids into the different poles of the cells. And by this time, you have one centriole, because we have a pair of centrioles, you have one centriole in one end of the cell and the other centriole in the opposite end. So you form a structure that is called an aster. So this is how mitosis looks like. So this is prophase, in which you have this nucleus with the condensed chromosomes, and you start having this disintegration of the nuclear membrane. And as you can see, these centrioles start going into opposite poles, and then they start extending these microtubules. And this is how this will look in an actual microscopic view. So you have these condensed chromosomes. And then during metaphase, in the equator of the cell, or this center of the cells, you align your chromosomes, your 46 chromosomes, into this equator in a horizontal manner. And then you have definitely a distribution of these centrioles in opposite poles of the cells, and then all of these microtubules elongate and attach into the kinetochores on both sides of the chromosomes. And then this will be called spindle fibers, and all this structure is going to be called an aster. And this is how it looks in the actual view of a cell with light microscopy. So you have here your metaphase plate. So this is the name for the alignment of the chromosomes in the equator, metaphase plate. Now during anaphase, you will have an enzyme that will cleave the sister chromatids apart at the centromere. And as this is happening, you have single-stranded daughter chromosomes migrating to each pole, because the microtubules start becoming shortened, and you have motor proteins in the kinetochores crawling along the spindle fibers, creating a physical force that is going to separate the chromosomes. And then the next phase, telophase, is when you already have separated your chromosomes, and then you have migration of the chromosomes towards opposite ends of the almost new daughter cells, and then you start reforming or making a new nuclear envelope around each cluster of the chromosomes into the two different cells. And then your chromosomes uncoil to chromatin, you dissolve the mitotic spindle, and then each nucleus will form a new nucleolus. So then this is what happens during anaphase. So again, you have these microtubules attached to this part of the sister chromatids, which are this, and then the microtubules start shortening, and then they're migrating toward opposite poles of the cells. And then as this is happening, you start making a new membrane, nuclear membrane, and then you have 46 chromatids, sister chromatids, in each of these new daughter cells, and then your cells starts forming this cleavage furrow that is going to pinch the cytoplasm of this cell, and then you form two new daughter cells, and then these chromosomes that were visible start forming chromatin. So this is how anaphase looks like in the light microscopy, and this is how the cells look like in telophase. So cytokinesis is the division of the cytoplasm into two cells, and it starts happening when you are at about to complete telophase, but overlaps a little bit cytokinesis. Now cytokinesis, it is achieved because there is this protein that is called myosin that it pulls over another protein that is called actin in the terminal ending or web of the cytoskeleton. And this pulling is going to create a cleavage furrow around the equator, and then the cell will eventually pinches into two. And like I said before, you have to distribute within the cytokinesis also equal number of organelles. So if you make mitochondria, just for saying one example, okay, so 10 mitochondria per cell. If you make rough endoplasmic reticulum, okay, so 5 for each, etc. It's not that it's that the real number, but like I'm trying to tell you is that you also have to have this equal distribution of organelles. So when does the cells will divide? So when they have enough cytoplasm for the two daughter cells, when they have replicated their DNA, and when they have adequate supply of nutrients, and they were stimulated by growth factors, and when you have cells that have died and there is an open space in between cells. So the cells regulate their cell division because of that. Now, the cells will stop dividing by contact inhibition. So as the cells are making new daughter cells and they feel contact with neighboring cells, they stop dividing. So one of the actual processes of stopping the cells from overgrowing is that by contacting neighboring cells. And also the cells, of course, if they don't have nutrients or growth factors, they won't divide, and they undergo contact inhibition, the cessation of cell division in response to contact with other cells. So we have for regulation of cell division a molecular timer in which you have several molecules that helps to control cell division. One of such molecules is an enzyme that is called cyclin, or several enzymes that are called cyclins. Now, the cyclins are not normally present during the entire cell cycle, but they form during interphase. And some of these cyclins are called cyclin-dependent kinases, in which you have these cyclins that are activated by the cell division cycles or phases, and they tend to phosphorylate other proteins. So there is this cyclin-dependent kinase complex control, in which when you replicate your DNA in your centrioles in the S phase, you start slowing down cell division because you have completed the replication of DNA and the centrioles by phosphorylating other proteins. And then your cells, your chromosomes, start condensing, breaking down the nuclear envelope, and then you form the mitotic spindle, and you attach this mitotic spindle into the kinetochores in the centromere of the chromosomes during prophase, and then you split the centromere and you separate the sister chromatids at anaphase. So these parts of the cell cycle, S phase, the replication of S phase and the centrioles in S phase, the replication of the DNA, condensation of the chromosomes, the formation of the mitotic spindle, and the splitting of the centromere are under the control of cyclin and cyclin-dependent kinases. And then the cells checkpoints, what they do is they prevent the cells from going into the next phase if the cells are not ready for it. So for instance, we have a G1 checkpoint. So this G1 checkpoint prevents the cells from going into S phase if you don't have materials enough for making a new DNA strand. Or you won't enter into this S phase if the cell goes into the G0 phase because it's a terminal cell. Now we have this G1 checkpoint controlled by cyclin-dependent kinase complexes. And then you have another checkpoint, which is the G2M checkpoint, in which once you have duplicated the DNA, you prevent the cell from going into mitosis if your cell has severe mutations. And then the cell can commit something or can make something that is called apoptosis or cell suicide. Sometimes the cells opt for not repairing the mutations in the DNA and rather they commit suicides. Now we have a third checkpoint at the transition from metaphase to anaphase that determines whether the cell can proceed to anaphase, and this will lead to the separation of the sister chromatids. Now the genes within the chromosomes are transmitted genetically from parent to the offspring. This is known as heredity. And in order for us to study the structure of the chromosomes, we have a technique that is called a karyotype, in which you put these chromosomes, the 46 chromosomes, into order by size. So the chromosome number 1, the pair of chromosome number 1, will be the largest, and then the 22nd chromosome will be the shortest. Now, out of the 23 pairs, we have 22 pairs that are called autosomes, that you have them no matter what is your gender. And we have the last pair of chromosomes that is called the sex chromosomes. That determines the genetic gender. So in females, we will have 2 X chromosomes and the absence of the Y chromosome. And then males, they have 1 X chromosome and 1 Y chromosome. And the X chromosomes and the rest of the 22 pairs of chromosomes that are called autosomes, they are homologous, they have the same size. But in males, the Y chromosome is way much smaller than the X chromosome. And actually the chromosome that determines sex, genetic sex, is the Y chromosome. Now when you have 23 pairs of chromosomes or 46 sister chromatids, you call this, you call it diploid cell or somatic cell. Now the majority of our cells are somatic cells, and we have these cells that are called haploid because they have only 23 unpaired sister chromatids, or 1N. And they have half as many chromosomes as diploid cells, or as the somatic cells. And which are the only 2 cells that have haploid number of chromosomes, or 1N, will be the sperm cells and the egg cells, which are germ cells. And when a sperm cell fertilizes an egg cell, you restore the diploid number, or 2N. So this is how a karyotype looks like. So you extract the chromosomes from a nucleus, you stain them, and then you take a micrograph and then you pair them up. So here we have all the autosomes, pair 1 up through 22nd. And as you can see here, pair 1, it has a larger size than pair 22nd. And then if you want to determine genetic sex, if you see two X chromosomes, this and no Y chromosome, this will be a female. And if you have an XY, this will be a male. Now, the actual location of a gene within a chromosome is known as locus. And we have different versions of a gene that are called alleles. And we have two alleles or more alleles depending on what trait you are talking about. Now, since we always inherit one set of chromosomes from our mom and another one from our dad, of course we will have two different versions. And the version that you can see in the person physically, that will be, or what the trait that this allele is coding for that you see in the person, this will be a dominant allele. And dominant alleles are represented by capital letter. And then you can have another allele that is called recessive. And this recessive allele is represented by lowercase letter. And when someone has a recessive allele and a dominant allele, that recessive allele that is coding for a trait is not going to be shown. And that's why it's called non-dominant or recessive. Now, usually dominant alleles codes for proteins that are responsible for a visible trait, and other ones, the recessive, they don't code for that protein and is not visible. So what are some examples of visible traits? Which is, by the way, a visible trait is known as phenotype, while your genetic sequence of DNA nucleotides in your genome, in your genes, is called the genotype. So some traits will be, for instance, for hair, will be curly versus straight hair. In your eyes, the color of your eyes will be, one allele will be blue, the other allele can be brown. And for instance, for that color of the eyes, for instance, brown trait or brown allele or version of a gene, it is dominant over blue. So the genotype, what it is, are the alleles an individual possesses for a particular trait, or the sequence of nucleotides within the DNA. Now, if the person has the same alleles, so if the person has both parents with brown eyes, the person will be homozygous for that allele. But if the person has one parent with blue eyes and the other one with brown eyes, the person will be heterozygous because he will have different alleles for that gene. What is a phenotype? It's an observable trait. How tall you are, the color of your skin, the color of your eyes, the texture of your hair, etc. Those are phenotypes. But genotype we cannot see because it's a sequence of DNA. And then in order for us to calculate the chances of getting a possible genotype and phenotype outcome, you can do it by using something that is a Punnett square. And in the Punnett square, you have plotting a single traits from each of the parents, and then you start making these combinations to see if the progeny will inherit a trait or not. So for instance, you can plot the possibilities of someone having cleft chin, which is a dominant trait, versus uncleft chin, which is a recessive trait. So this is how the actual Punnett square looks like. So you have a big square with 4 subdivisions in which you put here your potential genes from mom's side. So this will representing the X cells, and this will be representing the genes from that side with the sperm cells shown in here. So as I said before, dominant genes are represented with capital letters, while the recessive genes are written with non-capital or lowercase, like in this case. So for cleft chin, we have dominant C from the mother side with this egg, and lowercase c for this one. And then in here for the sperm, we have this sperm cell with capital C for cleft chin, which is the dominant allele, and then this lowercase c for the recessive allele. So what are the chances when you have these egg cells and these sperm cells combining? So when you have two dominant Cs, you will have a homozygous cleft chin. And then when you have heterozygous, because you are combining this dominant C from the sperm and this non-dominant or this recessive C from the egg cell, you will have heterozygous. So you have big C, little c, and then you still have a cleft chin in the progeny because you have one dominant coming from the sperm cell. What about in this situation in here? Well, it's the same, because at some point you have this cleft chin gene or allele coming from the mom, from the egg cell, and then you have a recessive coming from the sperm cell. So in here you have still, just like in this situation, big C, little c, and then you have heterozygous cleft chin, but since the big C or capital C codes is dominant for cleft chin, this progeny will have cleft chin. And what about when you have recessive trait of cleft chin coming from the egg and coming from the sperm, like in this case? So you will have homozygous recessive for uncleft chin. So in this case, this will be this case. So then what are the chances when you have this set of combinations for someone who to have cleft chin? So 3 out of 4 or 75. And what are the chances of someone from not having cleft chin? Will be 1 out of 4 or 25%. So that's how you calculate in single trait for these alleles. Now, genetics is more complicated of that, because many of these traits are coded from more than one gene. Now, what is the importance of learning this? Well, because sometimes the parents can be healthy and they can be heterozygous carrier for hereditary diseases, in which when they have a dominant trait for being healthy, they still have another gene or another allele that codes for someone to have a disease. And then you study those genetics, and then the person can have an advisory of whether the progeny can have a possibility of having a genetic disease or not. Now we have multiple alleles, codominance, and incomplete dominance. So all the genes that are found within an entire population will make a gene pool. And then since you have multiple alleles, more than 2 allelic forms more than 2 allelic forms will form a gene. And we have the example of 3 alleles for the ABO blood types. So if someone has IA and IB, the person will have codominance. Like in this case, the person will have AB blood. But the person can have also these I alleles, which are lowercase, that are basically non-dominant because they are recessive. And then in this case, the person have the possibility of having a different blood type. So for instance, if one of the parents has this lowercase I and it combines with this big case I and B, the person will have B blood type. And if the alleles of someone who has a lowercase I combines with a capital I A type, the person will have an A type of blood. In this case, this is non-dominant. Now you can have also incomplete dominance, in which someone might have a dominant allele and a recessive allele, and the person will show an intermediate type. So it's not dominant completely, but it is not recessive. Example will be when someone has a trait that is called familial hypercholesterolemia. And like I said before, most of the traits that we have in our body are made of polygenic inheritance. Poly means many. So there may be a gene in chromosome number 19 that it is contributing to a trait, and similarly there might be at the same time a chromosome in a gene in chromosome number 18 and 5 and so on that contribute to a single gene, a single phenotypic trait. And then these are harder to trace because many of these genes at different chromosomes contribute to the disease or the trait, and then sometimes it's very complicated to trace what is going on. So example of polygenic inheritance is eye color, skin color, and some forms of cancer. And then we have something that's called Pleiotropy, in which one gene itself can produce multiple phenotypic effects. So it's kind of at some point the opposite of polygenic inheritance. So in this case, you can have many phenotypic traits affected by a single gene. Example of this will be alkaptonuria, in which you only have the mutation on one gene of chromosome 3, and this blocks the breakdown of tyrosine, and you end up with different effects in the body, as we will see next. So this is polygenic inheritance. So we have genes at different locations in the genome, like genes 1, 2, and 3. This is just a representation that affects a single phenotype, which is eye color. And then you can have pleiotropy, in which a single gene can produce effects on different areas of the body. So one of the phenotypes will be someone having a darkened skin. Another one will be when the person has darkened sclera in the eye, or their urine becomes darkened upon standing, because tyrosine cannot be broken down. And also we have sex linkage inheritance, in which the traits are carried on the X and or the Y chromosome, but more than likely will be the X chromosome. Okay. Now the Y chromosome hardly will have any of this. Y chromosome, as I said before, it is determinant for genetic sex. And then one example of sex linkage inheritance will be someone who has a problem to seeing colors. So this will be color blindness. So we have a recessive color blindness allele on the X chromosome. And this will tend to affect more men than women, because men only have one X chromosome. So whenever they inherit a mutated gene for color blindness from the mom, the male will always be colorblind. But the daughter might not be, because the daughter has two X chromosomes, one coming from the mom and one coming from the dad. If the dad doesn't have the mutation but the mother does, then she will be colorblind. But if only she get the color blindness trait either from the mother or the father, and then the other one is normal, she will be a carrier. So this is just to illustrate the problem. So here you have this recessive trait for color blindness, which is the mutated one. And then in here you have this for normal color vision with capital. This is dominant. So if you put these two, the female genotype will be big C, little c. She will have normal vision because she only has a mutation on this, the recessive mutation. And the dominant prevents this from being expressed. But when it comes to males, when he inherits this color blindness gene coming from the mom and the Y chromosome coming from the dad, he will always be colorblind because he doesn't have the other normal chromosome for compensate. And this female is going to be a carrier, although she has normal vision. Now about RNA. So the RNA is another nucleic acid. It's called ribonucleic acid or RNA. And this RNA is long linear polymer, but it is much smaller than the DNA. And it can have between 100 to 10,000 nucleotides or bases. And we have two types of RNA that are important for protein synthesis. So we have messenger RNA, we have ribosomal RNA, and we have transfer RNA. Now, RNA is not a double helix, it's a single strand, and ribose sugar, it is replaced instead of deoxyribose sugar that is found in DNA, and uracil replaces thymine as a nitrogenous base, and RNA functions mainly in the cytoplasm. Now, the process of making proteins is known as translation, and translation is basically making a copy of a nucleotide but in another language, in the language of amino acids. So we have three main parts of, or three main structures that participate in translation. So we have the messenger RNA that makes this copy of the information coming from the DNA and bring it into the cytoplasm. So messenger RNA has a protein cap that it is the recognition site for the ribosome so that the ribosome can start making the proteins. And then the transfer RNA or tRNA will bring individual amino acids to add it into the growing chain of proteins. And this transfer RNA has a region that is called an anticodon that binds to the three nucleotides that are found in the messenger RNA. And this sequence of messenger RNA that codes for these amino acids is known as codon. Now, the ribosomes are located either free in the cytosol or attached to the ER or the nuclear envelope. And once they bind to the messenger RNA, they attract the tRNA charged with the amino acids and they start elongating the proteins and making the proteins. So, messenger RNA begins with something that's called the leader sequence. And this will act as a binding site for the small ribosomal subunit that you have either free in the cytoplasm or attached to the rough endoplasmic reticulum or to the nucleus. And then it will have a large subunit that is going to attach to the small subunit. And they will form like a little hand that helps to trap the messenger RNA, like if it was a ribbon, and then the messenger RNA will be passing in between the fingers of that little hand, and then it will start making these proteins. And the protein is being made when you have a sequence within the messenger RNA that is called AUG, which is the start codon. And then from there, you'll have the elongation of the chain of amino acids until you complete your protein. Now, all the proteins begin with amino acid that is non-methionine, because methionine is coded from this codon, AUG. Every 3 nucleotides on the messenger RNA, which are known as codon, will code for an amino acid. So AUG codes for making 1-methionine. Now, for the transfer RNA, it's the smallest of the RNA molecules, and it will coil on itself, making a structure that looks like an L. And in one end, you will put 3 nucleotides or anticodons that are going to match the messenger RNA codons, and then at the other end, the tRNA is going to bind to that specific amino acid that it was coded in the messenger RNA. And the tRNA will get these amino acids from the cytosol into what we call the free amino acid pool. And then as this tRNA is bringing these amino acids into the growing chain of polypeptides, you start using ATP. So this is how the transfer RNA looks like. So in one end has this area that is the charge area where you put the amino acids. And then in here, it's called the anticodon. This anticodon binds to the sequence of messenger RNA, and it's going to be specific for whatever amino acid is coded within the messenger RNA. So what are the 3 processes of making a protein? Initiation, in which you start with your AUG, the start codon, you put methionine, and then you start elongating your polypeptide by adding amino acids together in peptide bonds. And then you stop the process of making the protein because you have completed the sequence. This is known as termination. So initiation happens at the leader sequence in the messenger RNA that binds to the small ribosomal subunit, and then you have the initiator tRNA, which brings methionine, and then you start having this large ribosomal subunit that joins the complex, and now is fully engaged to start pulling this ribbon or these sequences of messenger RNA. So after you have done that, and start adding these amino acids one by one, you are elongating your sequence, your polypeptide. The process will continually repeat until you form the specific protein. And within this messenger RNA, you will have this stop codon that helps the ribosomal RNA from binding into the messenger RNA, and then you don't add more amino acids, and then you finish or terminate the formation of more protein. And then the ribosome will dissociate into the 2 subunits. Now, when you're translating to make a copy or many copies of proteins, you have to be very fast and kind of very accurate. So you form a series of ribosomes in line that is called polyribosome, that can be 10 to 20 ribosomes at once, and then you start making these proteins, and you can produce over 100,000 protein molecules per second. So this is a polyribosome. In these blue color, blue dots, this is an electron micrograph. You can see the ribosomes, and then see how each of these ribosomes are elongating these polypeptide chains, and in these will be the ones in green color, making thousands of proteins at the same time. And all these red red chain that you see here will be the messenger RNA that is being read or translated by these ribosomes. Now, what happens once you make the protein or the proteins that you needed to make? Well, the proteins can be packaged and stay within the cell, or some of them, they can be exported. So the proteins that are headed for the lysosomes, that are going to stay within these little vesicles inside the cell, or the ones that are going to be secreted, will be made on the ribosomes that are attached to the rough endoplasmic reticulum. And then from here, the cells will be modified and packaged into a transport vesicle that is going to go from the ER, rough ER, into the Golgi apparatus. So what is then the process of protein synthesis? Is when you activate a gene, then you make a messenger RNA through this making of the messenger RNA inside the nucleus of the cell, and then the messenger RNA will go from the nucleus into the cytoplasm to bind to ribosomes, and then you will start translating. Now, the process of making messenger RNA out of DNA is called transcription. And this happens within the nucleus. So this is it's called the central dogma of biology. So out of DNA, you make a copy of messenger RNA into what we call transcription. And it happens in the nucleus. And then this messenger RNA will exit the nucleus and it will make proteins. And this will be called translation. And it happens in the cytoplasm, either in free floating ribosomes or in ribosomes attached to the rough ER or in the nuclear membrane. So this is how then this messenger RNA is exiting the nucleus, which is this, through the nuclear pores, as you can see. And then this messenger RNA reaches the ribosomes with the two subunits, the big or the large subunit and the small subunit, and they form like a little hand when they attach the small and the big subunit, and then they grab this chain of messenger RNA and start reading its codons or its 3 nucleotides. And then these codons will bind to anticodons that are found in this tRNA, and this tRNA will make a cycle, so it will bind on this end the amino acids that are found in the pool of amino acids in the cytoplasm. And then as they're doing that, they spend ATP, and then they bring it into the growing chain of polypeptides to make a protein. So the direction of the ribosome movement will be from this part of the messenger RNA into the other one, and then the growing of the polypeptide chain is going to be in the opposite side. And then that's how you start making your different proteins. And then once you make the proteins, you can put them into the rough endoplasmic reticulum. You can modify the proteins, and then they can go into transport vesicles that are going to reach the Golgi apparatus in order for either to put it in vesicles as lysosomes or to export it out of the cells. So this is then the review of peptide formation. So here you have a DNA double helix. And out of this DNA double helix, you copy the sequences to make a protein, making this messenger RNA. So if you take out from this DNA this sequence, which is TAC, CGC, etc., you make a messenger RNA. As you can see here, now you have uracil. So AUG, which will be the codon for methionine, will be the start or the initiation of translation. And then this anticodon on the tRNA, which is this, binds to the UAC because it's complementary. A will bind to U, U to A, and then G with cis, and then it brings methionine. And then this part of the messenger RNA for the next gene, which is CGC, will have the opposite sequence, which is GCG, and then the anticodon will code for alanine, and so on. So every amino acid has sometimes one codon or more than one, and then they code for a specific sequence within the polypeptide chain. Now the proteins are processed in the following way. So protein synthesis is not finished when you form the amino acid sequence or the primary structure. You have to also form a secondary and tertiary structures and sometimes a quaternary structures by the help of proteins within the cytoplasm that are called chaperone proteins. These chaperone proteins will pick up the new proteins and help them to fold into their appropriate shape so that the proteins will be completely functional. Now, if you remember in the previous chapter, we mentioned that a misfolded protein will be tagged for destruction by the proteasomes. And these chaperone proteins then helps to prevent this from happening. So they fold specifically the primary sequence of the amino acids into a secondary and a tertiary structure, and then some of these tertiary structures will fold and well, not fold, but make an assemble that it will form that quaternary structure. So when does the proteins will leave the cells and when they will stay on the cells? So when the proteins are made on the free ribosomes in the cytosol, will be likely to stay in the cell. But if a protein moves from the rough ER, is made initially in the rough ER, and then it goes into the Golgi complex, more than likely the cell will be going outside of the cells. And then the Golgi apparatus, what it does is put some sugars into these proteins, so it will have like a little stamp so that the proteins will know where to go. So the proteins assembled in the ER surface will go through this series of endoplasmic reticulum cisternae through the pores that they have. And then the ER also will modify the proteins by what we call post-translational modification, in which you try to stabilize the proteins with bridges of sulfur, that is called disulfide bridges, or you add carbohydrates. And then when the rough ER is finished with the proteins, it will pinch off a bubble that it is coated with clathrin protein, and then this helps them to form like a little vesicle that is going to go and detach from the ER and will carry the protein now into the Golgi apparatus. Now, once the vesicle coming from the ER reaches the Golgi apparatus, it will unload the proteins into the channels of the Golgi apparatus. And then the Golgi will start modifying further this protein by adding carbohydrate chains. And as it's doing that, the glycoproteins are going to be transferred from certain areas of the Golgi into another ones that are farthest from the ER, and then you will end up with a finished protein. Now, some Golgi vesicles will become lysosomes, and other ones will be secretory vesicles that will go first into the plasma membrane, will fuse into the plasma membrane, and then will be released from the cells through exocytosis. So this is how the proteins are processed then. So you have the formation of proteins here within the ribosomes on the rough ER, and then they will pass through pores in this cisternae, and then when it reaches the area that it is close to the Golgi complex, they pinch off these little buds, and then these buds come and fuse with the first cisternae of the Golgi complex, and the Golgi complex keeps modifying these these vesicles that are coated with clathrin, and then as they move from the area that it was closer to the ER, the rough endoplasmic reticulum, and they move away, they will form either lysosomes that stays on the cell or secretory vesicles that comes and fuses with the plasma membrane by exocytosis. And this is all for this chapter. Let me just tell you the key points for these chapters 3 and 4. So the first key point is that the phase of mitosis in which the chromosomes align the cell's equator or the midline will be metaphase. And then one thing that I didn't mention in the previous chapter, chapter 3, is that when you are breathing faster and deeper, you eliminate more carbon dioxide from the body than normal breathing. And then this will affect diffusion. How? Well, most carbon dioxide will diffuse out of the blood. So when you are breathing deeper and faster, the effect on carbon dioxide is that more carbon dioxide will diffuse out of the blood. So these are a couple of points. Now let me tell you the other 2 points. So another key point is that the cell undergo mitosis to form 2 daughter cells, each with the same number of chromosomes as the original cells. And then the process of making proteins from messenger RNA is known as translation, and the phase of mitosis in which the chromosomes align in the cell's equator, as I told you before, is metaphase. Now let me tell you the other key points. So many proteins in the cytosol that specify or that speed, the regulation of metabolism are enzymes. So that's another key point. So enzymes are proteins that speeds chemical reactions, and we already saw that before, because these enzymes are known as catalysts, if you recall. Now another key point is that what happens when a cell has enzymes but there are no cofactors. So what will be the effect? Well, the effect will be that the enzymes won't work without those cofactors. So then this will be all the key points, and I will see you in the next video. Bye-bye.